Can one tablet a day keep the cancer away without surgery and without chemotherapy? Australian news reports a man suffering from late-stage lung cancer is being treated with a single pill a day. That and more on today's episode of Bad Geno. Welcome back to Bad Geno, bringing you the latest advancements in cancer genetics. Before we begin, nothing in this video constitutes medical advice. Please seek help from trained and licensed medical professionals for your healthcare needs. Last week, Australian news reported that a man was being treated for lung cancer with just one tablet a day. It's been hailed a miracle drug because lung cancer has a survival rate lower than colon cancer, with an 18.6% 5-year survival rate, according to the American Lung Association, compared to 64.5% for colorectal and 89.6% for breast cancer. Or to put it bluntly, more than half of patients diagnosed with lung cancer, they die within the first year of being diagnosed. Note that these are statistics from the US. But these statistics make this case in Australia all the more remarkable. The success of this treatment has been attributed to personalized medicine technologies. That is, the ability to detect the specific cancer type and tailor match that cancer to the right treatment. In this case, one tablet a day. Now, the patient was an Asian man diagnosed back in 2020 with advanced lung cancer at 44 years of age. He's a non-smoker, he's healthy, and he exercises, which is why he was caught completely by surprise. He was not expecting the diagnosis to be cancer. But now, after the treatment, he looks great. In fact, he says he doesn't feel like a patient, which is the best kind of outcome, I guess, you can hope for such a serious type of cancer. Again, the success of this diagnosis comes down to detection. How can you tell what type of cancer this is? And the way to do that is to sequence the DNA of the biopsy. A piece of cancer tissue is biopsied or cut out, and this tissue is sequenced in a machine that maps out the DNA. Then the DNA is analyzed for irregularities. In this case, it shows an EGFR exon 19 deletion. EGFR is the name of the gene or the section of your DNA. Exon is the subsection of that gene, in this case, exon number 19. And the deletion means the part of the DNA in this section it's missing or it's been deleted. Now, this is called a variant. And these days in Marvel movies, everyone's heard of that word variant. In DNA, a variant is simply a change or an anomaly in your DNA sequence. In fact, you can Google this variant. You can type in EGFR exon 19 deletion, and what you'll find are scientific papers written on this specific variant or change in your DNA. Interestingly, one of the papers in this results outlines the exact treatment given to this patient in Australia. So there you go. It's all on Google. Why is this important? Well, cancer is a disease of your DNA. And so detecting cancer involves looking at your DNA code for clues, for indicators of changes that might have caused the cancer. Doing this helps doctors to identify the cancer, but also to identify the right treatment for the patient's cancer. Normally, this would be surgery followed by chemotherapy. But in this case, neither were prescribed because looking at the DNA sequence of the cancer cells for this patient gave doctors the confidence that this was a type of cancer that didn't need surgery, that didn't need chemotherapy, that could be treated with this one specific drug. In this case, one pill a day. So let's look at this treatment. Tagrisso. Tagrisso is the brand name on the box. Osimertinib is the proper name for the drug, which is a series of tablets that you can swallow. According to the American Cancer Society, this is classed as an EGFR inhibitor. Reading the explanation. Osimertinib is a class of medications called kinase inhibitors. It works by blocking the action of the abnormal protein that signals cancer cells to multiply. This helps stop or slow the spread of cancer cells and may help shrink tumors. So what this drug does is it tells the cancer cells to stop growing. It blocks a chemical messenger or enzyme called tyrosine kinase. And usually these messengers tell the cell to grow or to divide, but blocking these messengers stops the cell from growing. Sorry, that was very technical. Think of it like stopping your postman from delivering your eBay packages. That kind of thing. It stops your eBay debt from growing. Anyways, the results of the treatment is amazing. Scans show that the tumors have shrunk and virtually disappeared, so it works. And again, without chemotherapy, without surgery, just one pill a day to swallow. And that's the treatment. Now let's look at the technology. Now this is the cool part. The pathologist, which is a kind of scientist that's trained to spot cancer, the pathologist, he or she does this by looking at x-rays, by looking at CAT scans, or by using microscopes to look at cancer cells that you've cut out or biopsied. But in this case, the pathologist or cancer detective uses DNA to spot the cancer type. This particular pathology lab in Australia uses the Ion Torrent Genexus integrated sequencer that's able to load 24 patient samples in one go. And it detects specific variants, that is changes in your DNA in particular locations. So you need to know which location you want the machine to focus on. There's usually a list of genes for it to concentrate on. And this is cheap compared to, say, looking at your entire genetic sequence or your genome. You're just looking at possible regions in the DNA, and this machine runs overnight. The next day, the software in this machine will report all the DNA variants it found in your list, and it will also suggest the possible targeted treatments or drugs available for these variants, and the clinical trials for the drugs that are still in the testing phase, but specifically target these variants that it found. You can think of it like a matchmaking exercise. You know, on the one hand, you need to know which type of cancer it is, which gene, which variant it is. But once you identify the cancer, but you also need to know what drugs or what treatment are available that target those specific cancers. Now, some of these drugs might be expensive, or they might not be available in your country, or they might be experimental, which can be a good thing because you can enroll in a clinical trial to test out maybe a new kind of drug that might be more effective than the current treatment options and might even be available for free. But either way, you need both the disease and the treatment. And that's what this machine called the sequencer does. It matchmakes them both. If you find this content useful, please help me out by liking this video and subscribing to this channel for more videos like this one. Thank you so much. I really appreciate it. Now, back to the video. Finally, let's look to the future of cancer detection, and it's called liquid biopsy, which is testing for cancer in your blood. It's considered by many as the holy grail of cancer detection. There's no need to cut out a biopsy or a piece of the cancer tissue, which can be very dangerous, very painful, and sometimes impossible to do because it's so deep inside your body. And the dream is to be able to detect cancer maybe even before it happens. The idea is for your blood to be drawn into vials and tested in a sequencer for traces of cancer DNA floating inside your bloodstream. Your blood, it pumps through your entire body, and it contains bits of cells from all parts of your body, including any potential cancer cells. In fact, some just use dry blood spots, meaning you prick your finger and you wipe a spot of blood on a piece of cardboard, which is again a lot less painful and a whole lot easier to do than an operation. The number one problem though with this technique is sensitivity. Maybe you have just a little bit of cancer DNA in your blood. So the sequencer that you use needs to be super duper sensitive to be able to pick it up. But the point is, this is the dream and everyone is trying to do this. Here's one example in Singapore. Here's another example in the UK. Everyone is trying to do this new technique called liquid biopsy for cancer. And it's very, very hard to do, at least currently. And we are not there yet. Most use a combination of techniques. They detect DNA, but also changes in your RNA and changes in your protein chemistry, that kind of thing. And that's why on paper, they'll say stuff like, we're trying to detect many, many kinds of cancer. They're casting a wide net to try to catch different types of cancers in your blood, but also because they don't want to put all their eggs in one cancer type basket, if that makes sense. So it's kind of like collecting Pokemon. You're trying to collect all of them, trying to detect all of them that's possible. And it's really, really hard to do. Another thing they try to do is to try to predict risk. That means they don't claim to detect the cancer, but they predict in a healthy individual who might develop cancer in the future. Meaning you're collecting blood samples from healthy individuals who might have a risk of cancer because maybe they have a family member who's had cancer before. And so you're just monitoring their blood levels, trying to monitor for changes and possible developments that might lead towards cancer. Now, there are other ways to doing this, not just testing your blood, but some try to use your breath. You blow into a device, and some use urine. I guess that's another form of liquid biopsy. And some even suggest building a toilet cancer detector. Very creative solutions. But blood is the most promising one that's most likely to see significant results before all these other forms of detection. Again, the problem with all these techniques is sensitivity. There are just so few cancer cells, if at all, in just one tiny vial of blood. And you can't exactly take buckets and buckets of blood from one patient to test. So it's a kind of needle in a haystack kind of problem. Personally, the one technology I'm most excited about is called Nanopore. Nanopore is a kind of sequencer that's able to read long lengths of DNA. And I've heard that Nanopore is developing a solution for this sensitivity problem that's very creative. Now I'm going to do a separate video on sequencing technologies covering Nanopore especially. So just know for now that in my opinion, I think these guys are headed in the right direction. Okay, so what are the conclusions about this news in Australia? Firstly, it is amazing. It is amazing that this kind of treatment is now available. Now it doesn't work for all cancers. It's very specific, just this particular kind of cancer, just this particular kind of variant. But for this one guy who got this treatment, it's made all the difference in the world. And the challenge is to identify other individuals, other people who might benefit from this specific treatment type. Secondly, we are entering into an era of personalized medicine. Personalized medicine means tailoring the treatment to the individual, tailoring the treatment to the type of cancer. And this is possible because the cost of sequencing your DNA has dropped so much. It used to be, like 100 million pounds 20 years ago to sequence just one person. But now it's like $1,000. So it's a lot cheaper comparatively to what it was before. It's still expensive, don't get me wrong, but it's made it possible. It's opened up the doors to more cancers being diagnosed more accurately. But having said that, it doesn't solve the problem of treatment because you have the diagnosis, but the treatments are still inaccessible, they're still experimental, and many of them are still very, very expensive, especially in countries like in Malaysia. But at least, the sequencing is getting cheaper, and this opens the door to more diagnosis, to more research, and to more drug discovery, reusing drugs maybe sometimes for different types of cancers. So yeah, these are very exciting times, and it's great news for this one guy who was able to benefit from this technology, just taking one pill a day. Thank you so much for watching. This has been the very first episode of Bad Genome. Please like this video and please subscribe for more updates on genetics in cancer and medicine. Thank you for watching. Take care and see you in the next one. Bye.